this is going to be a review for you. It's anatomy. So we'll try to cover the anatomy down to physiology tonight. Mr. Ziminski, you're holding me up now. Okay. All right. So when we have a new organ system, we want to look at its function. All right, so what do the kidneys do?
Filter the blood.
Well, they filter the blood, don't they? Now, this is not like the spleen filtering the blood. When we say the spleen filters the blood, what is it doing?
Taking out expired red blood cells.
It's taking out old and or damaged red cells and platelets, isn't it? Okay, so in this sense of the kidneys filtering blood, it's a different kind of filtering. Because the blood, it's filtering, but all the blood cells still stay in the blood. All right, so it's filtering basically the plasma portion of blood. That's what it's doing. All right, and so in that filtrate is going to be everything, not just metabolic waste, but nutrients are going to be in there, water's going to be in there, all of that. But we don't want to flush that down the drain, every bit of it. We want to get the nutrients back. We need those. We want most of the water back. What we want to get rid of is the waste material that the body doesn't need or shouldn't have. And we'll add to that things that the body needs to get rid of.
Is that why you have efferent arterioles and then the actual convoluted ducts? Is it the clean blood goes out through the efferent arterioles and then the waste goes through the...
No.
All right, well, it's worth a shot.
No.
Okay.
Not quite like that. All right, we're going to find out that the kidneys have a major role in water and electrolyte balance. We're going to find out that renin, which is an enzyme, which is part of the renin-angiotensin system, comes from the kidney. We already know about this hormone, don't we? Erythropoietin. Do you remember where it came from? Well, it comes from the kidney. All right. And the kidneys also produce this hormone right here, calcitriol. So this has a lot to do with vitamin D absorption. All right, so that's what the kidneys are doing. Now, what's left in the filtrate that passes through the tubular system of the kidneys is urine. And that's going to pass down these tubes, the ureters, into the bladder, this temporary storage organ, and out of the bladder it's going to pass through the urethra to the outside. So here's the whole setup pretty much here, as we've seen. The two kidneys, the two ureters, and the urinary bladder. This happens to be a female, so you notice that the urinary bladder is the most anterior organ in the pelvis. Behind it is the uterus, and behind that's the rectum. I showed you a film from an IVP in lab. This is one film of a series of films that make up a study known as an IVP, or intravenous pyelogram. And this particular film here is pretty, in stark contrast, because you can see virtually the whole thing. You can see the kidneys outlined right here on both sides. And the contrast medium now is being excreted into the calyceal system. You see the minor calyces right here? In the major calyces, renal pelvis, and down the ureter into the bladder. And both sides are showing up sort of equally here. So this is a later film in the series, And it's basically showing you two normal kidneys here. All right, we know that the kidneys are posterior, and they lie under ribs 11 and 12, and that they are retroperitoneal. So peritoneal cavity here, and so this is parietal peritoneum here, and the kidneys are behind that, so they are retroperitoneal. They have a thin capsule. They're encased in renal fascia, deep to which is lots of adipose tissue. The kidneys are cushioned in a lot of fat. And the combination of the capsule attached to the fat, attached to the renal fascia, attached to the parietal peritoneum, holds the kidneys basically in place. All right, we looked at the anatomy of the kidney. The kidney has two major zones, an outer cortex and an inner medulla. So this lighter area here is cortex. The medulla is staining a darker brown and showing the renal pyramids in contrast here. These are not quite shown as well here. This is a diagram based on this. So this is cortex. Of course, the capsule is out here. All of this is medulla. And these individual darker brown things are renal pyramids. These are the minor calyces right here at the end of the pyramids. These little projections at the end of the renal pyramids here are the renal papillae. Okay, so what we have up here in the cortex, we're going to find out, are the renal corpuscles and convoluted tubules. And down here in the medulla, we're going to have loops of Henle and collecting ducts. Okay. Now, I don't think I told you about these before, but these little things between the pyramids here, those are called renal columns. All right, so here are the minor calyces, as we said, leading into major calyces here, leading into the renal pelvis, and that's continuous with the ureter. We talked about the blood supply to the kidney, so we'll go over that briefly. It's outlined for you right here, but here's the renal artery, segmental artery, interlobar artery, arcuate arteries. Arcuate arteries arc over the tops of the renal pyramids here, and they give off multiple branches up into the cortex here. These branches are called interlobular arteries or cortical radiate arteries, and it's off these arteries that the afferent arterioles come, and that's what these little branches are that are not labeled on this diagram. So we go over here with aorta, renal artery, segmental, interlobar, arcuate, cortical radiate or interlobular artery, leading into the afferent arterioles, which lead into the capillaries. The glomerulus is the capillary component of the renal corpus. Now, remember I told you that the kidneys are unique in that these capillaries here are drained by another arteriole, not a venule. Most capillaries, in fact all capillaries elsewhere in the body, are drained by venules, but this one is drained by an arteriole, and as I told you, there's a reason for that we'll explain later. And this arteriole then goes into capillary networks, either peritubular capillaries around the cortical nephrons, or vasorecta around juxtamedullary nephrons, loops of Henle.
The long loops.
All right, and so coming out of those capillaries now, we do have veins. So we have cortical radiate veins or interlobular veins. And so let's go back over here. Here are the cortical radiate veins or interlobular veins, which drain into arcuate veins, which drain into interlobar veins. Let me go up here. Here's an interlobar vein, which drain directly to the renal vein. So we've left out a couple of vein names that might go with arteries here. That's just the way that the setup runs. And so eventually we get back to the inferior vena cava. Now what about the nerve supply to the kidney? Autonomic nerve supply to the kidney is sympathetic. As far as we know, there is no parasympathetic innervation of the kidney. So sympathetic inputs to the kidney adjust the rate of urine flow, and therefore it will adjust the glomerular filtration rate that we'll talk about later. So sympathetic input is going to have a role in controlling blood flow and blood pressure in the nephron. We will also find out that sympathetic discharge is one of the causes of release of this enzyme right here, renin, which is going to initiate the renin-angiotensin system that we'll talk about later. All right, so we come down the ureters to the bladder. So the bladder is a temporary storage organ. It's very muscular. Its muscle has a particular name. It's called the detrusor muscle. Now these ureters that come down, they're lying on the psoas major muscles. They drop over the pelvic rim, and they don't just run right into the top of the bladder. They lie in the posterior aspect of the pelvis, down to the pelvic floor, and approach the bladder from behind and low, and enter the bladder obliquely. And this is where they open into the floor of the bladder right here. The control of flow of urine out of the bladder is through two sphincters. The setup is similar to the anal sphincters and how they work. So the internal urethral sphincter is down here low in the bladder at the bladder neck. So this one here, internal urethral sphincter, is involuntary. It's autonomically controlled. And I'll show you that later on when we talk about voiding the micturition reflex. All right? The other sphincter is down here in the pelvic floor. So there is a ring of muscle around the urethra right here called the external urethral sphincter. Now this muscle up here is smooth muscle, autonomically controlled. This muscle down here is skeletal muscle. It's somatically controlled, cortically controlled. So this is voluntary. See, so when you get ready to void and the bladder contracts, this sphincter relaxes. But what keeps the urine from coming out?
External.
Because this one's still constricted right here. You have conscious control over this, and so you relax this sphincter when it's convenient to empty the bladder. This happens to be a male right here, so we've got the prostate between the bladder and the pelvic floor right here. But you can see the urethra is running right through the prostate.
I have a question. Like I said, but you know when you have the urge to go and then you don't get to go right away and then it goes away, why does that happen? Because
you override, you're holding the urine in here by keeping this constricted, and over a period of time, those urges will abate. But the urine's going to keep collecting in there, and the whole reflex is going to start again, and you're going to feel the urge again. You may be able to control it again, and they'll abate, but eventually, you're going to have to go. And if it's not convenient, you're going to go anyway. Because the pressure that builds in the bladder will overcome the strength of this sphincter right here.
And females have both internal and external?
Uh-huh. Here's the internal sphincter in the female right here. Same place as the male. And the external sphincter is in the same place because this is the pelvic floor right here.
We just don't have a
It's
prostate.
just that the urethra is shorter in a female. So you have the same setup. Just the anatomy is different. All right, so the functional unit of a kidney is the nephron. The nephron. Now, you know by now that nephrons are microscopic. You can't look at a kidney and see the nephrons. All right, because in each kidney, there's over 1 million nephrons in there. And so they're all bunched up and they're tiny. All right, so, but this is a diagram of one, and this diagram is stretched out this way. In the kidney, they're all bunched up tight like this. It's stretched out here so you can see it. And actually, there are two kinds of nephrons here. So to orient you, these vessels right here are the arcuate vessels. This is arcuate artery and arcuate vein. So with that in mind, then, what region of the kidney is this up here?
Cortex.
Cortex, and then this down here would be
Medulla.
medulla, wouldn't it? All right. So here comes an interlobular artery, or cortical radiate artery. And here is one branch of that right here.
Afferent
And
arterial.
so this is an afferent arterial. Afferent means going toward or going to.
Into the
Where is it
glomerulus.
going?
The glomerulus.
It's going over here to this renal corpuscle. So this whole thing right here that I'm circumscribing is renal corpuscle.
And the glomerular capsule is inside the renal corpuscle, or are they
No,
the same
no.
thing?
Now, the capsule is this outer part of the renal corpuscle. Right
Okay,
here.
all right,
This is the glomerular capsule,
Capsule.
or Bowman's capsule.
okay,
This is the capillary.
right, which is the glomerulus.
This is the glomerulus.
Right.
All right. The subcapsular space is the space between the capillary and the capsule.
Okay,
That's this space in here, around it.
okay. So if we were looking
Renal
at this
corpuscle is the whole thing.
possibly on a lab test, looking at a model, and there was a pointer right to the round area, would we call it a renal corpuscle or a glomerular capsule?
Okay, well,
'Cause I'm
if the pointer is right here, what you going to call that?
a renal corpuscle,
If the pointer is here, what you going to call
glomerulus.
Yeah.
Okay.
Of course, it depends on how the question's worded, but,
Okay.
all right? Now, this is where the blood is filtered. So it comes in here through this capillary and out here. This is the efferent
Through the
arteriole.
vasa recta.
All right. So the blood's going to stay in the bloodstream. Okay, so we're actually filtering the plasma, aren't we?
Yes.
What's dissolved in plasma.
Water.
For the kidney to work, things have to be in solution. The kidney can't work with dry material. So things have to be dissolved in solution. All right? And so that's what's going to show up in the fluid that accumulates in this space here, which is heading down this tubule.
The
right here.
proximal convoluted tubule.
And this is the proximal convoluted tubule. All right? And so it comes to here. Now, from here, we go out of cortex into medulla. And so
Loop of
now
Henle.
we're in the loop of Henle.
Mhm.
Then we come back to the cortex,
To the distal,
and we're in the distal
Convoluted tubule. to the
And notice that
common
it takes
collecting
a
duct.
turn up adjacent to the renal corpuscle. You see it? This is distal convoluted tubule right here. And then it heads over to the
common collecting duct.
duct.
Yes, which goes down
All right,
to
so
the
this is
renal
just
pyramid.
one nephron draining into this collecting duct. Another one could be here, another one here, another one here, as you see. All right. Now, this collecting duct is in the medulla, isn't it? Just like the loops. All right. Now, where is this urine going?
To the renal papilla, and then
It's
the
going to
minor
drip out
calyx.
the ends of those renal papillae into the minor
Calyx.
calyces.
To
isn't
the
it?
calyceal system, out to the renal pelvis, down to the ureter.
Right.
What you said.
Sorry.
So we keep talking about it.
Now, there are two kinds of nephrons shown here.
Cortical and
This is cortical
juxtamedullary. Short loop.
This is a juxtamedullary nephron.
Long loop.
Now, what's the big difference?
The loop length. Plus your
The loops.
vasa recta is in the loop of Henle more, so is that just a diagram?
No, the vasa recta are associated with the loops of Henle of juxtamedullary nephrons.
Okay. So up there is just the paratubular capillaries? Okay.
All right. Now, they're called juxtamedullary because they are
Proximal
next
to
to
the
the medulla.
medulla.
Juxta means in
Proximal.
proximity to. So you'll notice that these renal corpuscles and the convoluted tubules are situated lower in the cortex. So about 85% of nephrons are this type, about 15% are this type. So why do we have two different types?
That does something different. It has to do with a type of filtration, correct?
No,
No?
this has a lot to do with the ability of the kidney to concentrate urine, as I will show you later on. All right, so, in summary, we got about 1.2 million nephrons in each kidney. There are two types, as we just mentioned. The renal corpuscle consists of those three components that we mentioned: the capsule, the glomerulus, and the subcapsular space. And the renal corpuscles are unique because their capillary is both fed by and drained by an arteriole, the only place in the body where you see this occur. All right. Now, let's look at the tubular system. What are the tubules for? For reabsorption of things the body needs to keep. Nutrients, water, things like that. Things that the body doesn't need will go on without being reabsorbed, or are only being just minimally reabsorbed. There are some things that the body needs to get rid of that don't necessarily come out in the filter. But the tubular system has the ability to secrete things into the tubular fluid and get rid of them, get them out of the body. So secretion basically is a backup to reabsorption. All right, so our tubular system then consists of the proximal convoluted tubule, and that's where we're going to reabsorb most everything. We're basically going to reabsorb all the nutrients there and most of the water. Then the loop of Henle. The loop of Henle is only involved with recovery of water and certain minerals, certain electrolytes, sodium, chloride, and some potassium.
Okay.
That's the only thing that happens there. The distal convoluted tubule does reabsorb some stuff, but it has the capacity to secrete things into tubular fluid. So by secretion, we mean things pass from blood into interstitial fluid, into tubular cells, into tubular lumen, and down the drain. All right, that's what secretion is. Reabsorption is the opposite. Reabsorption is taking things out of tubular fluid, into tubular cells, into interstitial fluid, into blood. So they're the exact opposites of each other. And of course, that tubular system ends at the collecting duct. All right. The capillary beds, we know that filtration occurs in the glomerulus. We know that it's fed by and drained by arterioles. These peritubular capillaries wrap around the convoluted tubules because they're going to absorb the things that are absorbed from the tubular fluid into the tubular cells, into interstitial fluid, into blood. Okay, so we need those nutrients, we need that water, it's got to be in the bloodstream. All right, the vasorecta, as I mentioned, are a specialized capillary network around the long loops of Henle and juxtamedullary nephrons. Later I'm going to show you their importance in maintaining the solute concentration of blood, because we're going to find out as that tubular fluid passes into those loops, there are going to be some massive shifts in water and sodium and chloride.
Is that because of diffusion?
If our blood reflected what's happening in the kidney down there, we'd be like this with the solute concentration of our blood. Okay. We couldn't keep it steady. It needs to be steady or relatively steady.
For homeostasis.
Okay. Was it on time?
Yeah, I think so, yes.
All right. Okay. So now let's look at the renal corpuscle in a little more detail. We got the capsule, glomerular capsule or Bowman's capsule. Now, there's epithelium involved here. The inside of that capsule is lined with an epithelium.
Is it simple squamous, or is it
simple squamous epithelium?
And is that... Okay.
But some of that epithelium is also covering the glomerular capillary.
Okay.
So we got this parietal visceral thing again. Remember how that goes?
Yeah.
So there was a space there. The capillary grew into it and pushed some of that parietal epithelial lining of the capsule ahead of itself. And so we're going to find out that the capillary is covered with a specialized epithelium.
1.2 million of them. Oh, wow.
We're going to find that the glomerular capillary is a certain type of capillary. Fenestrated. Remember the types of capillaries? Right, we'll show you that in a moment. We're going to talk about the filtration membrane. Now, this subcapsular space is just that space between the capillary and the inside of the capsule. All right. And notice that this is a subcapsular space, not the subscapular space. The subscapular space is under here, okay? Under the scapula.
You're not referring to me, Dr.
The names have been
removed.
changed to protect you. All right. Now let's look at this thing. So this gives us a little more detail, doesn't it? So here is our afferent arteriole, And here is the capillary here, but now it's covered with this visceral epithelium that's the same epithelium as the lining. It's just a different name. Now here is parietal epithelium, here it's visceral epithelium. Here is a piece of a glomerular capillary with its fenestrations. Now why would there be fenestrations in these capillaries here?
Because the density of the solute, I know I'm saying that wrong, but because has to do with the fact that you've got the plasma proteins and that's what it's filtering, or this plasma.
There's a lot of filtration going on here. All right,
So you need larger pores.
The fenestration facilitates that. All right, so that's good. Now, so this right here in yellow, you see, is going to be this epithelium here. But now we're seeing some detail here. Because this epithelium that covers the capillary has little extensions called foot processes. They're podocytes with their foot processes. So here is an electron photomicrograph of this right here. So you see here, this is the visceral epithelium with its podocytes and little foot extensions. You see the little extensions here, little foot processes. Now, if we drew a diagram of this, this is what it would look like over here. This is the filtration membrane over here, diagrammatically represented. So, here's the capillary. Here's a fenestration. Here's another fenestration. Here is the basement membrane of the capillary. And in yellow is that epithelium that's covering the capillary. And so these are foot processes here, stuck down on the capillary. But between those processes, there are little slits called filtration slits. Now this is a bigger view of that. Same thing. So here is the capillary with its fenestrations. Here's the basement membrane of the capillary. Here are the foot processes of the podocytes, and here are the filtration slits. So basically, this filtration membrane is multi-layered, isn't it? And the little openings are smaller as you leave the capillary. In other words, you got to pass through a filtration slit. You got to go through the basement, to the fenestration. You got to go through the basement membrane, and these filtration slits are smaller than the fenestrations. Now, where are we when we get out here? We're in the space, in the subcapsular space, aren't we? This is where this fluid is collected. And it's going to head down the proximal convoluted tubule. So let's look at the tubule. So we're leaving now, and we're in this proximal convoluted tubule. What's going to happen in this proximal convoluted tubule? The main thing is reabsorption. What are we going to reabsorb?
Nutrients,
water. Nutrients. Okay, some ions and some proteins. Now you say, I thought that the plasma proteins were too large to get through the filter. Most of them are. But some of them are small plasma proteins, aren't they? So sometimes some of the small ones can get through and pass into the proximal convoluted tubule. But if they do, they are promptly reabsorbed by the proximal convoluted tubule. So we shouldn't be passing any protein in our urine, should we? Under normal circumstances. Sometimes some of those little plasma proteins get hung up in the filter, and if they do, they're phagocytized. Get rid of them. All right? Now, the epithelium in the proximal convoluted tubule is cuboidal, and it has microvilli. What does the presence of microvilli tell you?
Oh, it's got enzymes.
Absorption.
More absorption,
More absorption.
because you're increasing the absorptive surface, aren't you?
So,
By those cytoplasmic extensions.
so this microvilli doesn't have the brush border enzymes. That's only in the small
it's just
intestine.
in the digestive tract.
Okay.
These microvilli are to increase the absorptive surface area
Okay.
of tubules. All right? Now, as I mentioned before, these reabsorbed substances are going to pass into the fluid surrounding the tubule. This is interstitial fluid. In the kidney, we commonly call it peritubular fluid. Peri means around, doesn't it? All right? And then into the capillaries, and of course, then we'd be in the systemic circuit, wouldn't we? And as I mentioned previously, secreted substances move in the exact opposite way. So reabsorbed things that are in tubules, they pass from tubular lumen into tubular cell, into peritubular fluid, into blood capillary. Secreted things that the body needs to get rid of that haven't been passed through the filter come out of blood into peritubular fluid, into tubular cell, into tubular lumen, and out. Now, so secretion is the movement of things from blood into interstitial or peritubular fluid, into tubular cells, and into tubular lumen. All right? But once it gets into the tubular lumen, removal of those materials from the body is called excretion. So there's a difference between secretion and excretion. So excretion basically is anything moving across an epithelial surface out of the body.
So is excretion then the reabsorption of the secretion, kind of, in layman's terms?
No.
No?
Excretion is getting
Just
rid of what's
excreting
been
it.
secreted.
Okay. Getting rid of what's been
Yeah.
secreted.
All right. Now, so right here is the epithelium of proximal convoluted tubular cells. Notice that it's taller and it has microvilli. All right, now, the loop of Henle. So as we leave proximal convoluted tubule, now we pass into the loop of Henle. So we're here now, like this. What happens in the loop of Henle?
Recovery of water.
Recovery of water
Concentration in urine.
and
Concentration
recovery of electrolytes. I should have put that in there. We are recovering some electrolytes too. I left that out. So we're recovering water and sodium and chloride and some potassium. The loops of Henle have an important role in concentrating the urine. All right. Now, we know that the loops of Henle have a descending limb and an ascending limb, and they have different functions. What we're going to find out is that the descending limb is permeable to water, but not to solutes. And the ascending limb is permeable to solutes and not to water. Later we'll show you how this works. All right, so we're going to pass. I mentioned this before in lab, that the loops have a thick segment and a thin segment. You can see that in the models. That has to do only with the height of the epithelium. So part of the loop has a low epithelium, part of it has a taller epithelium. It has nothing to do with the tubular lumen.
But does that have to do with what it's permeable to? No?
Not necessarily.
Okay.
Well, yes, because the descending limb typically has one kind of epithelium and the ascending another kind.
Okay.
But the main thing for you to remember is that the descending limb is permeable to water and not to solute. The ascending limb is permeable to solute and not to water.
Okay.
We'll see how this works later.
Okay.
All right, out of the loop, we're back in the tubular system. And we're in the distal convoluted tubule. So now we are here, like this. So the distal convoluted tubule begins where the loop ends. It's confined to the cortex. Remember I told you that a portion of it passes very close to, in fact, right up against the renal corpuscle. There's a reason for this, as we shall see. The distal convoluted tubule can absorb and secrete things. When it's reabsorbing water, we're going to find out that it only reabsorbs water under the influence of a certain hormone called aldosterone that we'll talk about later. And so the distal duct is going to end at the collecting duct. All right, now, let's talk about where that distal convoluted tubule butts up against the renal corpuscle.
So it never enters, because the way the book looks, you can't, it's
It does
deceiving. It
not
almost
enter.
looks like it
It does
goes
not enter.
back into it. Okay.
This is the way it looks right here. So this is the distal convoluted tubule right here, and this is the renal corpuscle, isn't it? All right. Now notice that this piece of distal convoluted tubule is snugged right up against the renal corpuscle. It's very close to the afferent arteriole and the efferent arteriole. All right. Let's identify some stuff that's in here. This figure is in your book, by the way. All right, so we know about these things, don't we? Afferent arteriole, glomerular capillary. This whole thing is renal corpuscle. This is the capsule. This is the subcapsular space. And this is proximal convoluted tubule, isn't it? All right, now let's identify some other things that are in here.
Okay.
These gray cells in here are called mesangial cells. They have a lot to do with constriction and dilation of the capillaries. So they can adjust flow through the capillaries, can't they?
Yes.
All right. So some of them are inside, wrapped around and amongst the twisted part of the capillary, right? And some of them are just outside. So these would be infracapsular or glomerular mesangial cells. These would be extracapsular. All right. Notice another thing. There are some specialized cells here in the wall of the afferent arteriole. These are called granular cells. This is where renin, an enzyme, comes from, and this is where erythropoietin comes from. All right? Now, notice that in this distal convoluted tubule segment that's next to the corpuscle here, there are some specialized cells in its wall. These are called macula densa cells. And they have a certain function. These cells are going to monitor the flow rate of fluid coming through the tubule at this point. And they are also going to monitor the cellular concentration of that fluid that's coming through the tubule at this point. We will come back to this later and explain why that's important. All right, but this whole thing here, this whole arrangement that we're talking about is called the juxtaglomerular apparatus. Juxta means next to or in proximity to. It's next to the glomerulus, isn't it? So it's the juxtaglomerular apparatus. And all of these things, these specialized cells in here, have a function, as we shall elaborate on a little bit later. Well, let's do it right now, actually.
All right.
What's going to happen here? We're going to monitor renal blood flow. Because if you look right here, this is a reflection, what's coming through this artery is a reflection of renal blood flow, isn't it? The blood flow is coming off the aorta into the renal arteries and eventually into these little arterioles here, isn't it? So if blood volume and blood pressure drops in the body, isn't that going to affect how much blood is flowing into the kidney? And therefore how much blood is flowing into afferent arterioles? So it's going to make a big difference in the way the body responds to that. So those macula-densa cells, as I told you, they're going to monitor the tubular fluid flow rate. They're going to monitor the solute content of that fluid, and namely that's going to be sodium and chloride.
Do they do that through specialized receptors?
No, these cells are specialized
Receptors,
right
okay.
here. These cells have the capacity to do this.
Okay.
So that's what these macula-densa cells are.
Okay.
These mesangial cells, we said, some of them are inside the capsule, some are external to the glomerulus, but they have the ability to regulate the glomerular flow rate because they can squeeze down on the arteriole or they can relax and let more blood flow in. The granular cells, as I mentioned, secrete renin and erythropoietin. All right, so that brings us now to the end of the nephron, and so we're in the collecting system now. So we're in the collecting duct. So this is where we are. We're here now, and so this fluid that's passing out of distal convoluted tubule in the collecting duct, we can now call this urine. But we haven't been calling it urine back in here, haven't we? Because things are changing. You know, its concentration and composition are changing because we are reabsorbing things out of it, we're secreting things into it, we're calling it tubular fluid. But by the time it gets here, we can call it urine, but it's got to pass down this tube. And on the way, we can adjust the concentration and composition of that urine even more. So this, what's coming down the collecting ducts here, is not necessarily of the same composition and concentration as what's stored in the bladder. When it drips out the end of a renal papilla, that's equivalent to what's in the bladder. But on the way down here, these collecting ducts have the ability to absorb water, they have the ability to secrete or reabsorb certain electrolytes like bicarbonate and hydrogen ions and so forth, as we shall see. All right. So we're going to adjust the composition of urine. We're going to adjust its final concentration in these collecting ducts. What happens in there? We're going to reabsorb some water maybe. We're going to recover some sodium, maybe get rid of some potassium. We can secrete or reabsorb hydrogen ions depending on the pH of the blood. We can secrete or reabsorb bicarbonate ions depending on the pH of the blood. Now, I'm introducing something that's not in your book. The very lowermost end of these collecting ducts and other sources are called papillary ducts. So in your notes, I think on page 6 or so, what page is that?
6.
Page 6, at the bottom, there's a little diagram that shows you where the papillary ducts are.
Okay.
It's not a different duct. It's the same thing as a collecting duct. It's just at the very lowermost end of it, there's something special that happens there. Okay, this is where urea is reabsorbed through these papillary ducts, the lowermost portion of the collecting duct. Your books don't mention papillary ducts, but other sources do. So it helps me to know this, because one of the things that the kidney does is it reabsorbs a good bit of the urea.
Mm-hmm.
Now, a lot of it passes in the urine, but not every smack dab bit of it. So it's normal to recover some, because it helps us to concentrate urine, as I'll show you later. And therefore, since we're reabsorbing some's going to be in the blood all the time, isn't it?
Mm-hmm.
Low levels. All right. So that gets you through the anatomy, basically, of the kidneys.
Okay.
So we'll stop here. Next time, we will delve into renal physiology.
I have a question, something I'm kind of stumped on. In the beginning of this,
Listen, folks. Quiet, just a minute.
this is something I'm stumped on. In the beginning, you said that there's very
I'm giving
little,
back tests tonight if you need to see it.
if any sympathetic, you said parasympathetic. You said it was mostly sympathetic nervous, not parasympathetic. But how can that just kind of, I guess it's, maybe I'm just not, I don't have a great understanding of the sympathetic and parasympathetic. It's just kind of blown my mind with all that going on. It seems like it would be parasympathetically controlled. All right. It's not. Okay. I need to look that up then.